prevent infectious diseases are given to millions of babies, children, adolescents, and adults. And it is critical that they are demonstrated to be safe and effective. Vaccines have prevented countless cases of disease and disability and have saved millions of lives. Ensuring the safety and effectiveness of vaccines remain a top priority. Vaccine development is a complex science. Developers need to ensure that the rigorous scientific and regulatory processes are followed in the development of any vaccines. Regulatory authorities conduct a thorough evaluation to determine the safety and effectiveness of vaccines. There are typically years of clinical trials needed to produce a safe and effective vaccine, with researchers testing multiple vaccine candidates as failure rates are generally high. This module aims to provide an overview of how vaccines are made, how long it takes, and to break down what the vaccine development process normally looks like. However, a comprehensive discussion on the technical information is beyond the scope of this module. In our previous session, it was briefly discussed how vaccines work. It works by mimicking the infectious bacteria or viruses that cause disease. Vaccination stimulates the body's immune system to build up defenses against the pathogens without causing the disease. The parts of the infectious organism that the immune system recognizes are foreign to the body and are called antigens. Vaccination exposes the body to these antigens. Some vaccines contain weakened versions of a bacteria or virus. Other vaccines contain only part of the bacteria or virus. Some vaccines contain only the genetic material for a specific protein and direct the body to produce a small amount of that protein. The body's immune system reacts defensively once it detects this protein. After vaccination, the immune system is prepared to respond quickly and forcefully when the body encounters the real disease-causing organism. Vaccines contain tiny fragments of the disease-causing organism or the blueprints for making the tiny fragments. They also contain other ingredients to keep the vaccine safe and effective. These latter ingredients are included in most vaccines and have been used in decades in billions of doses of vaccine. Each vaccine component serves a specific purpose, and each ingredient is tested in the manufacturing process. All ingredients are tested for safety. All vaccines contain an active component or the antigen, which generates an immune response or the blueprint for making the active component. The antigen may be a small part of the disease-causing organism like a protein or sugar, or it may be the whole organism in a weakened or inactive form. Preservatives prevent the vaccine from becoming contaminated once the vial has been opened if it will be used for vaccinating more than one person. Some vaccines don't have preservatives because they are stored in one-dose vials and are discarded after the single dose is administered. The most commonly used preservative is 2-phenoxyethanol. It has been used for many years in a number of vaccines and is used in a range of baby care products. It is safe for use in vaccines as it has little toxicity in humans. Stabilizers prevent chemical reactions from occurring within the vaccine and keep the vaccine components from sticking to the vaccine vial. It can be sugars like lactose or sucrose, amino acids such as glycine, gelatin, and proteins, recombinant human albumin derived from yeast. Surfactants keep all the ingredients in the vaccine blended together. They prevent settling and clumping of elements that are in the liquid form of the vaccine. They are also often used in foods like ice cream. Residuals are tiny amounts of various substances used during manufacturing or production of vaccines that are not active ingredients in the completed vaccine. Substances will vary depending on the manufacturing process used and may include egg proteins, yeast, or antibiotics. Residual traces of these substances, which may be present in a vaccine, are in such small quantities that they need to be measured as parts per million or parts per billion. Some vaccines also contain adjuvants. An adjuvant improves the immune response to the vaccine, sometimes by keeping the vaccine at the injection site for a longer time or by stimulating local immune cells. The adjuvant may be a tiny amount of aluminum salts like aluminum phosphate, aluminum hydroxide, or potassium aluminum sulfate. Aluminum has been shown not to cause any long-term health problems, and humans ingest aluminum regularly through eating and drinking. A diluent is a liquid used to dilute a vaccine to the correct concentration immediately prior to use. The most commonly used diluent is sterile water. Most vaccines have been in use for decades, with millions of people receiving them safely every year. As with all vaccines, every vaccine must go through extensive and rigorous testing to ensure it is safe before it can be introduced in a country's vaccination program. Because vaccines are given to healthy people to prevent illness, the rigor to which safety and testing are held is higher than for treatments given to people who are already ill. Scientists develop a rationale for a vaccine based on how the infectious organism causes disease. The scientists then conduct laboratory research to test their idea for a vaccine candidate. Sometimes, this testing occurs in animals. This is considered the research and discovery stage. Once a scientific finding is thought to have practical applications, in that it may be feasible to develop a vaccine candidate based on that finding, the research moves forward. Each vaccine under development must first undergo screenings and evaluations to determine which antigen should be used to invoke an immune response. Before a vaccine can be tested in people, researchers perform additional laboratory research and testing in animals to obtain information about how the vaccine works and whether it's likely to be safe and work well in humans. These tests are known as the preclinical phase. An experimental vaccine is first tested in animals to evaluate its safety and potential to prevent disease. If the vaccine triggers an immune response, it is then tested in human clinical trials in 3 phases. When the developer is ready to begin studies in humans, they compile the results of their laboratory and other preclinical testing, as well as information pertaining to the manufacturing technology and the quality of the vaccine, and submit this to the regulatory authorities such as the FDA in the form of an investigational new drug application. The authorities' evaluation includes an assessment of the preclinical data and a determination whether these tests were conducted according to good laboratory practices. It also conducts an assessment of the product, its quality and safety, and the technology to manufacture it to determine whether it is reasonably safe for testing the vaccine to move forward in people. Studies conducted in people are known as the clinical development stage and typically occur or cover 3 phases under the oversight of regulatory authorities. Clinical trials are conducted according to plans that reflect the authorities' considerable expertise in clinical trial design. These plans are called protocols. Vaccines intended for children are generally tested first in adults with a step-down clinical development program to children and infants. The phases of the studies may progress sequentially, but it is also not uncommon for the phases of development to overlap. In phase 1, the emphasis is on safety and generally includes 20 to 100 volunteers who haven't been exposed to the disease being studied and who are generally otherwise healthy. These studies are used to determine whether there are adverse reactions with increasing doses and, if possible, to gain early information about how well the vaccine works to induce an immune response in people. In the absence of safety concerns from phase 1 studies, phase 2 studies include more people where various dosages are tested on hundreds of people with typically varying health statuses and from different demographic groups in randomized controlled studies. These studies provide additional safety information on common short-term side effects and risks, examine the relationship between the dose administered and the immune response, and provide initial information regarding the effectiveness of the vaccine in its ability to generate an immune response. Standardized and validated tests are used to evaluate the immune responses. These vaccine studies typically also include a control group consisting of people who may receive regulatory authorities approved vaccine, a placebo, or another substance. People receiving vaccine under the study are compared to people in the control group. In phase 3 clinical trials, the vaccine is generally administered to thousands of people, and the study generates critical information on effectiveness and additional important safety data. This phase includes additional information about immune response and compares those who receive the vaccine to those who receive a control such as a placebo. For example, the number of cases of disease in the vaccinated group is compared to the number in the control group to see whether the vaccine reduces the incidence of disease. These studies also provide information about the vaccine's safety, including the identification of less common side effects. When the results of all these clinical trials are available, a series of steps is required, including reviews of efficacy and safety for regulatory and public policy approvals. Officials in each country closely review the study data and decide whether to authorize the vaccine for use. A vaccine must be proven to be safe and effective across a broad population before it will be approved and introduced into a national immunization program. The bar for vaccine safety and efficacy is extremely high, recognizing that vaccines are given to people who are otherwise healthy and specifically free from the illness. Regulatory authorities make its decision based on its analysis of the benefits and risks for the intended population who will receive the vaccine, as well as the diseases to be prevented. Its scientific team works collaboratively to evaluate all of the scientific data and information included in the licensure application and makes the determination whether to approve a vaccine or not. A typical team is comprised of physicians, chemists, statisticians, pharmacologists or toxicologists, microbiologists, experts in post-marketing safety, clinical study site inspectors, manufacturing and facility inspectors, and labeling and communication experts. Further monitoring takes place in an ongoing way even after the vaccine has been licensed or approved for use. There are systems to monitor the safety and effectiveness of all vaccines. This enables scientists to keep track of vaccine impact and safety even as they are used in a large number of people over a long time frame. These data are used to adjust policies for vaccine use to optimize their impact, and they also allow the vaccine to be safely tracked throughout its use. Once a vaccine is in use, it must be continuously monitored to make sure it continues to be safe. It is important to note that a vaccine is a drug. Like any drug, vaccines have benefits and risks, and even when highly effective, no vaccine is 100% effective in preventing disease or 100% safe in all individuals. Most side effects of vaccines are usually minor and short-lived. For example, a person may feel soreness at the injection site or experience a mild fever. Serious vaccine reactions are extremely rare, but they can happen. Although the vaccine development process and regulatory authorities' evaluation are rigorous and comprehensive, there is still a need for ongoing surveillance of vaccines after approval to identify uncommon adverse events or long-term complications that may occur, and sometimes to monitor effectiveness. In certain cases, the regulatory authorities may require the manufacturer to conduct post-marketing studies to further assess known or potential serious risks. These studies are sometimes called Phase 4 of development. In summary, vaccines are one of the most important tools in combating infectious diseases, and developing one that is safe, effective, and affordable are incredibly difficult to make and must go through rigorous safety procedures before they can become widely available. On the average, it takes 10.7 years to develop a vaccine. General stages of development cycle of a vaccine include an exploratory stage, a preclinical stage, the clinical development, and a regulatory review and approval. Clinical development is a three-phase process. During phase 1, small groups of people receive the trial vaccine to assess its safety, confirm it generates an immune response, and determine the right dosage. In phase 2, the clinical study is expanded and vaccine is given to people who have characteristics such as age and physical health similar to those for whom the new vaccine is intended in order to further assess its safety and ability to generate an immune response. In phase 3, the vaccine is given to thousands of volunteers and compared to a similar group of people who didn't get the vaccine but received a comparator product to determine if the vaccine is effective against the disease it is designed to protect against, and to study its safety in a much larger group of people. Most of the time, phase 3 trials are conducted across multiple countries and multiple sites within a country to assure the findings of the vaccine performance applied to many different populations. Many vaccines undergo phase 4 formal ongoing studies after the vaccine is approved and licensed. Every vaccine must go through extensive and rigorous testing to ensure it is safe before it can be used among humans or introduced in a country's vaccination program. Vaccine development undergoes the most rigorous process to ensure safety, immunogenicity, and efficacy. In an era of increasing vaccine hesitancy, we hope that this module helped in the